We're going to be looking at enzymes, and essentially these are a type of catalyst, specifically a biological molecule that is going to be acting as a catalyst, and they are going to be used as such. Since they are catalysts, they are going to be speeding up chemical reactions. So let's say you had some milk, you get some lactose in that, and our body wants to be able to get some energy out of that lactose. It has to be able to break it down, and that can be done by breaking it into glucose, or when it gets broken down, it gets broken into glucose and galactose. So you take a lactose disaccharide, and this bond that is holding it together needs to be broken. So we have a particular enzyme that is able to break that down. But in order to break that bond, you're going to have to input energy. Anytime you break bonds, you got to put in some amount of energy. You'll get some out as well, but you got to put some in to get that bond to break. And in order to do that, one way to do it is to use heat. However, the heat needed to break that bond would also destroy a cell. And so what we have instead is that we don't have to heat it up a whole lot to break that bond, though that would work, it would kill our cells. Instead, we use an enzyme to do that. So enzymes are protein catalysts. Again, they are biological molecules that act as catalysts, and they are very specific to the substrates or the things that they're going to be working on. They speed up the reaction. They themselves don't get changed by the reaction, so we say they don't get consumed. They come out of the reaction the same way they went in, and they do this by lowering the activation energy. So we mentioned the energy required to get a bond to break. So if we're breaking bonds for this particular reaction, say breaking down lactose, it's going to require a certain amount of energy. So if you imagine, if we start off with our lactose and we want to break it down into those monomers, we're going to break it apart. You've got to put in, you start at a certain amount of energy, and you've got to put in some energy, and that's where you actually get the breaking of the bond. So here's where we'd have the breaking of the bond, and then we'd have some bonds reforming with those the H's and OH's. They're picking up from water, but this is the high energy point in the reaction, and it requires a certain amount of energy to get there. Too much energy for cells to handle in terms of heat energy, but what can be done instead is a catalyst can be used where there is less energy needed. So the same thing will happen, the bond will break, but it happens at a lower energy level. There's less energy required to get it to happen. That is what an enzyme does, is it essentially grabs the substrate and makes it easier to break apart than what would be needed if you were just trying to break it apart with, say, heat or just allow the reaction to happen. It happens at a lower energy level or less activation energy needed. So how do they go about lowering this activation energy, or basically how do they make it easier to break those bonds? The enzymes themselves have amino acids, and the amino acids have these specific R groups to them, and those, whatever the R groups happen to be, are going to be used to actually break or weaken the structure within the substrate itself. So a few ways that this can be done, but one can be to put it in a particular orientation, so it can actually bind with this substrate and put it in a particularly, face it in the right direction. It can also transfer electrons to it, so that's an option that some enzymes do, is that they'll use they'll be called oxidative enzymes, but essentially they're able to use the transfer electrons to or from the substrate in order to actually weaken the structure of it, make it easier to break those bonds. Or they can add or remove hydrogen ions, and these are called hydrolytic enzymes, but that again would cause the substrate itself to become less stable, and it'd be easier to break those bonds. Therefore, lower activation energy for the reaction to happen. So here's what the process would look like, drawn out in diagrams. So an enzyme has a particular spot called the active site, and this is where it binds with the substrate. So you can imagine the substrate could be something like lactose, and the enzyme is going to have a particular spot on it. And remember, this enzyme is a protein, right? So it's a big long chain, where they just draw them here as a glob here, but it is a big long chain that's folded in on itself, and somewhere in that conformation there is room for the substrate to fit. That is called the active site. When the substrate and the enzyme bond, the active site itself is binding to the substrate, and the enzyme substrate complex itself is sort of changing it to fit that. So we call this the induced fit model. It's not like a lock and a key where neither the lock nor the key really changes shape all that much. It's kind of more like your hand grabbing onto something. So let's say you're trying to take the lid off your pen or something like that with one hand. Your hand would have to sort of take the shape of the pen in order to grasp it properly and able to cause a change within that pen and pen lid and break it apart. That's something similar to what happens with the enzyme. Is it fits onto the substrate, or the substrate fits onto it, and it changes the sort of shape. They sort of fit together, and all the different polarities within the substrate and the enzyme itself are going to match up in a particular way, and then it is able to loosen that those bonds that are holding the substrate together, and is able to break it apart. Now, some enzymes do build things together as well, right? It's not always breaking things apart, so they can build things as well. But again, that will take some breaking of bonds, and so the enzyme helps with that and reforming new bonds to put them together. So this process can go over and over and over again, so it is a cycle. And if we're breaking these substrates apart, we can call it a catalytic cycle. Again, say imagine taking a substrate like sucrose, and an enzyme can grab onto that sucrose and break it apart. And again, this is a process where we're breaking that bond apart. We are using water here, so it is hydrolysis, or we're splitting the substrates apart. We're breaking the substrate up, and we're getting these two new monomers back from that. In this case, this disaccharide, we're getting these monosaccharides out of it. The enzyme is left unchanged, so it can therefore do the cycle again and again and again. So occasionally you need something to work with the enzyme to get it to actually become a functional enzyme. And so these are come in two different varieties, or we'll talk about two of them anyways. If they're an organic molecule, and you may have heard of coenzymes, they are an organic molecule that sort of binds with the enzyme, and then the enzyme is then going to be functional. Or it could be a more smaller piece of something like an ion. And if that is the case, if it is a particular ion that needs to bind onto the enzyme to make it functional, it's called a cofactor. So cofactors are generally smaller, simpler particles like ions. Larger organic molecules, if they're working with the enzyme to make it functional, it's called a coenzyme. So the rate of enzyme activity can be changed by heat. And so enzymes are going to be doing their thing, but their three-dimensional structure is really important, and that is really dependent on the temperature that they're in. If the temperature changes, their three-dimensional structure can change shape, and so we refer to this as being denatured, and then they can't do their job anymore. It doesn't fit into the substrate. The substrate doesn't fit into the enzyme anymore because they now have a different shape. So heat, either high or low, depending on what the enzyme is built for, which temperature it actually works at, either high or low and temperatures can actually cause it to not work as well or stop completely. pH is also going to change how, remember the enzyme is a protein made up of these amino acids, and so if it's going to be changing anything like the ionization of them, it may gain or lose more hydrogen ions depending on what the pH of the surrounding is, and that is going to change their shape as well. And if this happens, so we can look at, here's the rate of reaction on the y-axis here, and we can see that as you change temperatures, a particular enzyme, so let's look at just one of them here. So this one right here has this sort of bell curve type graph of its efficiency, and so where it is most effective is going to be for a particular temperature. So this is outlining human enzymes, and as you'd imagine, human enzymes probably work best at human body temperature in terms of where they function the most. It gets too hot, they don't work very well. It gets too cold, they don't work very well. If you are a bacteria that lives in a really hot place, then you are probably going to be full of enzymes that are going to be able to function at high temperatures, because that's where they need to be functioning. If the temperature gets too hot again, they don't function, or even if it gets too low, it will not function. And we can take advantage of things like that in some of our technologies like PCR. Same thing happens for pH, is that again, if you are an enzyme that works in a stomach environment with a low pH, you want to make sure that your conformations are such that it works well in that particular pH environment. Whereas if you are an enzyme that has to work in a high pH environment, again, its optimal performance will be right around that high end range. If you took trypsin and you put it into your stomach, it would no longer work, because by then the activity of that enzyme is so low, it would essentially be not working at all. So they're very much affected by temperature as well as pH. Also, the amount of substrate that the enzyme is working on is going to have an effect as well. So we can see here that looking at rate here, so how fast the reaction is happening, as you have more of the substrate that the enzyme's acting on, you're going to see an increase in the rate of reaction. So concentration goes up of the substrate and the rate of reaction is going to increase to a point. And so at some point it actually does become saturated, and so the enzyme is as busy as it can be. Whatever enzymes you have there, they're constantly grabbing onto the substrate and reacting with them. And so at that point, so it doesn't matter, so let's say that's about here, you hit the plateau, and adding more substrate doesn't matter because you've saturated your enzyme, so it can't work any faster than that. This graph also shows the reaction without the enzyme, and so increasing concentration does increase the rate of reaction, but as you can see with the enzyme, that rate increases much more quickly with having the enzyme present. Now also in terms of rate of reaction with the concentration of the actual enzyme itself, it would be a more linear relationship. As you increase the amount of the enzyme, the rate is going to increase pretty much proportionally. But with substrate concentration, you actually see this sort of very, very steep increase at the beginning because there's going to be a certain amount of enzyme present, and as long as you add more substrate, those enzymes are going to be able to work faster and faster until they are saturated. So these enzymes themselves, really important to regulate them. We want them to be working sometimes and not working other times, and so we have lots of different mechanisms to get this regulation to happen. Competitive inhibition is one, and this is where we have an inhibitor that competes for the enzyme's active site. So where the substrate is supposed to be landing on the enzyme, there's another thing that could land there as well. So we call this a competitive inhibitor, and again, it could land into that active site, and therefore the actual substrate can't, and so the reaction is going to be slower. So you can imagine if these substrates are trying to get to the active site, but instead there's this other thing that is not the substrate that also fits there, it'll stop those substrates from binding, and then the substrate itself is not going to be reacted. It's just going to sit there. There's nothing happening with it. So this is called competitive inhibition, when the active site of the enzyme itself gets blocked by something else. A quick example of this would be methanol is poisonous. So it's a type of alcohol that is really poisonous. Ethanol is also poisonous, but not to the same extent. But methanol poisoning, what can happen is that it gets broken down, and we get these toxic substances in our body. So if you can actually stop it from breaking down, that is not a bad idea. So ethanol actually acts as a competitive inhibitor for the enzyme that breaks down methanol into these toxins. And so if someone is suffering from methanol poisoning, one of the options is to give them ethanol, because it will actually act as a competitive inhibitor on the enzyme that would otherwise be taking methanol and making these toxic compounds. So it blocks that enzyme and gets it to not break down the methanol. Another example is how penicillin works on the actual reactions that bacteria are trying to carry out. So again, they act as a competitive inhibitor, and they stop essentially these the building of these cell walls within bacteria. The bacteria are trying to carry out this reaction to build their cell walls. Penicillin gets in there and acts as a competitive inhibitor, stopping it from happening and giving your body a chance to kill the bacteria itself before it can replicate more so. Non-competitive inhibition is, again, you're controlling that enzyme, but it is not using the active site to do it. So there is allosteric regulation, or inhibition specifically, that we're going to talk about now. This is non-competitive. It's not going for the active site, but there is somewhere else on the enzyme where these things can land, these regulatory sites or allosteric sites, And when this inhibitor, lands on that allosteric site, it changes the enzyme in a way that causes it not to work anymore. All right, so you can imagine we've got our enzyme here. The substrate's supposed to fit into the active site, but if this non-competitive inhibitor, so this is a allosteric inhibitor, lands on this allosteric site here, it changes the shape of the enzyme, and the enzyme cannot bind to the substrate and therefore will not be able to carry out the reaction with the substrate. So one example of this, and this is not an intentional thing that our body would do, but we have this cytochrome C as part of our metabolism processes, and it has a spot in it where potassium cyanide, or the cyanide itself, is able to bind to that enzyme. So the enzyme is supposed to be carrying out its job, helping us produce energy. Cyanide can bind to it and cause it to not be able to do its job anymore. Therefore, we don't produce enough energy, and that's not good. There's also this thing called feedback inhibition, and it could work with a allosteric inhibitor, but essentially the inhibitor itself is the product of the process that produces it, and will feed back onto that process to shut the process down. So essentially something gets created that then acts as a allosteric inhibitor on the enzyme that made it. So if we look at this process here, so you can imagine there's this process being carried out in several steps, but in the end there's this end product, and the end product itself is the inhibitor. And so the end product can go back and bind onto the enzyme that was actually used to make it, shutting down the process itself. And that seems kind of counterintuitive. Why would you want to make something that then shuts down the product that actually makes it? But essentially that's really, really important in biology, because we only want to make a certain amount of it. And once you have enough of it, it can go back and shut down the process itself until you don't have enough, and the process will start up again. So this is called feedback regulation. And we can see the difference in these two types of inhibition. Competitive inhibition tends to have a higher, remember we talked about the rate of reaction. So competitive inhibition, you have the direct competition of the substrate and the actual inhibitor fighting for that site, and so they tend to have a higher rate of reaction. So we'll inhibit it to some amount, but you can see that the competitive inhibitor is going to lower the rate of reaction, but it still gets to a relatively high amount, a relatively high rate. A non-competitive inhibitor, you don't have this direct competition between the inhibitor and the substrate, and so it's actually able to shut the reaction down at a lower rate of reaction. So allosteric regulation, we have this inhibition where it shuts down the enzyme, and then there's the exact opposite of that, where the enzyme might not be actually viable yet. It might not be able to do its job until something binds to it and then allows it to become an active enzyme or increase its activity even. And so we can have an inactive enzyme where a activator, so not an inhibitor, but an activator, binds to a particular, and again, this is called an allosteric site. And so we have this activator binding to the enzyme itself, and then the enzyme actually becomes active. It can actually do its job. It can then bind to, in this case, it can bind to this particular substrate here and then do its job. So this is an activator, very similar to an allosteric inhibitor, but instead of inhibiting the enzyme, it activates it. So one kind of example of that would be actually oxygen is kind of an interesting one on hemoglobin. When hemoglobin is this protein that we have that is binding to oxygen and is allowing our red blood cells to carry it throughout our body. And when oxygen binds to hemoglobin, it actually makes hemoglobin more able to bind to more oxygen molecules. And so when one binds, it acts as an allosteric activator, allowing even more oxygen to bind to that hemoglobin, which is kind of neat because it's the allosteric activator as well as the substrate.